section is always going to be in this manner. The right and then the left side. And then the right and the left side for reaching each visual field, right? So when you are told about hemifield, it's not like this in the middle, up and bottom, but it's always like this, right and left side. That's what I'm trying to say. So anyhow, but we can also still talk about quadrant anopias. And anytime you have anopia or anopsia, the term, it means blindness, right? The eye or the visual centers do not see that information. So anytime that you are looking into these eye fields, this is the patient's head, right? Patient's right and patient's left, and you see it this way. What makes things a little bit complicated is that it's not that these collections from the right and the left side of the eye in each eye is not going to be completely decussating. Some of these fibers will decussate, some will stay ipsilateral and continue their trajectory in that manner, right? This is the one that I was trying to say. In the visual field, right eye, you have hemisections as the right and left, and the same hemisections on the visual field of left eye as the right and left, right? And then these quadrants could be there, and this tiny little spot that we see is the blind spot. What happens is that if you can understand this whole thing, it's then not a big deal. What this is trying to say again, our eyes, all these photoreceptors are not standing in a planar sheath-like structure, right? They are not on like a line in one line. What happens is that because of our eyes are basically ball-like structures, what happens is that if I'm on this side of this ball-like structure, what I can see is the opposite side like that. And if I am, as a cell, sitting here, what I'm going to see is the opposite side like that. Okay. This is what it's trying to talk to you about. So you are going to have a temporal retina, and it's going to see the left visual hemifield for the right eye, and then you are going to have the nasal retina is going to see the right visual hemifield for the right eye, right? What happens is that these temporal retinal fibers trying to see the left visual hemifield in my right eye are going to stay ipsilateral. They are not going to cross over at the optic chiasm, and they will reach to the lateral geniculate nucleus on the ipsilateral side. Whereas my nasal retinal fibers trying to see the right visual hemifield in my right eye, right? They are the ones, when they get to the optic chiasm, they are going to cross over and go to the opposite side. So both eyes will do the same. Nasal retinal fibers will cross over at the optic chiasm. And the naming of this area, from my retinal layer all the way to the optic chiasm, this area is the optic nerve. And when I have these decussations or staying on the same side happens, it's the optic chiasm, this X-shaped area. From optic chiasm to the thalamus, this section is the optic tract. And then from the thalamus itself to all the way to my visual cortex, then now those things are called optic radiations. So you have optic nerve, optic chiasm, optic tract, thalamic nucleus, and then you have the optic radiations. Right? The optic radiations, we'll look at them a little bit, yes.
So if you have a split in the optic chiasm, like or a hemorrhage, are you not going to be able to see from these part of your visual fields?
Yeah, I'll show you the examples. It's coming in the slide. So yeah, there are specific things that can happen just at the optic chiasm. So you'll have a very typical, you're not going to see the sides, right? Because the fibers coming from your nasal field, we're supposed to see outside basically. They are the ones crossing. If you push them or hit them right at the optic chiasm, this person cannot see either side, right? And then it's going to be always that they will hit the wall, door frames and things like that. Right? As I was saying, optic radiations could follow two separate projections, and I'll show them to you what it means. Earlier again, when we were looking into some sleep lectures or hypothalamic nuclei lectures, what we said was that there's this extra geniculate pathway, right? Because the geniculate pathway basically tries to go to the lateral geniculate nucleus of the thalamus. That's the visual information. But that extra geniculate nucleus, those extra fibers, need to go to other places to let them know about the light information as well. So the geniculostriate pathway, right, relays that information to the lateral geniculate nucleus and then continues to the occipital cortex, and then you have these extrageniculate pathways, right? They will come to the superior colliculus, and then they can also get to the pretectal area. Remember when we were looking into tectum tegmentum around the brainstem level, that this pretectal area, we kind of talked about that at that point. What this one is trying to show, right? By the time you get to the lateral geniculate nucleus of the thalamus, it's going to have these 6 layers. It's still part of the neocortex communication areas. What's going to happen? It's going to communicate these specific layers from the lateral geniculate nucleus exactly to the occipital cortex, 6 layers as neocortex. And it's going to have these retinal ganglion cell layers and magnocellular cell layers. So parvocellular, magnocellular differentiation. And as you can see, each level of cells will take contralateral, ipsilateral, etc. That information like that. So it's trying to combine the 2 eyes for you. By the time you get to the occipital cortex, it's not really 2 separate visions that you are seeing, but you can see them as one. The thing that I was trying to say, we said from the thalamic nucleus back to the occipital cortex, we have these optic radiations, and they can take 2 separate trajectories. They can either go through these things called Meyer's loops, and they will pass through the temporal lobe to get to the occipital cortex. And then you are going to have these secondary loop, right? It's going to go towards the parietal lobe to reach up, down, I guess, to the occipital cortex. So what happens is, these Meyer's loop passes through the temporal lobe. These are the superior fibers pass through the parietal lobe. They don't have a specific name, really. What happens is, these superior fibers will end at the upper portion of that calcarine fissure that we looked at before. This fissure was similar to that, like, almost like a, what else we had? Like a Sylvian fissure, or we have the central sulcus, etc. So it's just a separation of the gyri in that occipital cortex. But wherever the fiber ends, it's going to determine whether you can see the lower or the superior portions of your visual field. We always talked about it like that for years, and finally in one book I found this figure, and I think it's like the best one that you can find ever, because it really gives you the idea like what we are trying to say. So if this is your visual field, right? Superior visual field, the macula in the middle, and then the inferior visual field. Who sees my superior visual field is really the inferior retinal fibers, right? Again, goes back to the idea. My eye is not like straight like this, but it's a shape like that. So anyone sitting here can see the ones on the top better, and the cells sitting up here can see the visual field at the bottom, right? So my superior retina sees the inferior visual field. The superior retinal fibers will come to my thalamic nucleus, and then from there they will continue towards the parietal lobe and then end up in the upper portions of the calcarine sulcus. So if you have a damage in the calcarine sulcus in the superior portion of it, what you are missing is that inferior visual field. The opposite is also true, right? The inferior retinal fibers will see the superior visual field. Once that information is collected, it's going to move to my LGN in the thalamus, and through the temporal lobe with the Meyer's loop, they are going to reach to my occipital cortex and end at the lower portion of the calcarine sulcus. So if I have a damage there, coming from the Meyer's loop, the visual field that I lose is going to be the superior visual field. This is going to be important for your patient, because a lot of the times when people have like epileptic seizures, of course it could start from any place, but most commonly those things start from the temporal lobes. When these epileptic seizures start from the temporal lobes, because the Meyer's loop passes within the temporal lobe, it's going to bother those Meyer loop fibers. And as we said, the Meyer's loop are the ones trying to see the superior visual field, right? During or right after that seizure happened, and seizure means that overactivity of the neurons, right? They keep functioning or firing action potential. Right after that, these people will have blurriness or kind of sort of transient blindness in their superior visual field. And it all comes back from the neuroanatomy of the thing. Another thing that we can tie this figure at the bottom, right? It also shows that, remember last hour we said, whatever you have in the macula or in the foveal pit, each photoreceptor is connected to a ganglion cell. So whatever they see is going to be perfectly mapped into the occipital cortex. That's why, even though you have this tiny little area, information-wise, collected from the center of your gaze, by the time you come to the occipital cortex, they will be devoting a larger, I guess, area to understand that central vision, right? Area number 17. And then as you go towards the periphery, 2 and 3, even though they are looking at a larger area, because we said all those photoreceptors are converged together, so the information is fuzzy, because I don't devote as much space to them in my occipital cortex. So this is the thing, goes back to all the, I guess, visual field defects that you can expect to find in patients. So there are certain typical ones. We will go through all of these, but there are typical ones that the board will tend to ask you. So I will, I guess, the questions that you can look at it together. So of course, if something happens, if there's a lesion on the optic nerve, that eye is going to be blind. And it's very straightforward, so you will not get a question like that, right? But that's what it is, basically. Whether they were going to decussate or they were going to stay ipsilateral doesn't matter, because you have a lesion right in there, so no one is going to be seeing anything from that eye. By the time you come to the optic chiasm right here, as we said, the nasal retinal fibers from both sides will be the ones crossing over, right, decussating. If you were to have a lesion right here, if you follow the fibers back again, this is the nasal field, but it was trying to see the periphery like that. So I'm not going to be able to see that area. And these other fibers were collecting the information from here, and they were trying to cross. What I was trying to see, the opposite side, so I'm not going to see that. So this thing is called bitemporal hemianopsia. As I said, anopia or anopsia, you can hear either one, is fine, which means it's blindness. Hemianopia, if you ever hear, it's always right or left. It's never superior or inferior, like that. And it's bitemporal because I'm not seeing my temporal vision, right? That's what it's trying to tell you. So bitemporal hemianopia. Tiny little thing can happen right here, not very common, but it could be just the fibers stayed ipsilateral coming from the right eye. But these fibers, if you follow them back, they were trying to see the left hemifield of my right eye. So I'm going to call that left hemianopia in the right eye. I'm just looking into anatomically where these fibers were coming from. Now, when I pass the optic chiasm, I'm in the optic tract right now. What happens over here, if you were to have a lesion in the optic tract, that is going to be now have a different name. Now it's going to be called homonymous hemianopia. Again, it's hemianopia, so it's on one side of my visual field. Homonymous means they are on the same side. It's either on the right or on the left side. And the easy way to remember which side that you are looking at, if you were to have these optic tract fibers damaged within the right hemisphere, the visual field you are losing is going to be left one, right? Left hemianopsia in the right eye. That one. So, left homonymous hemianopia. What I'm trying to say, this is not the, okay, I'm very distracted, I'll just take it. If this happens in the right hemisphere, right, we are saying that it's left homonymous hemianopia. We are not trying to say, right, it's just we are trying to determine the visual field is left, not like it's the entire left side is missing, okay? So only the left hemifield in both eyes. And because they're on the same side, I call this homonymous, you know, the same side on the both parts. The same thing could have happened right here, right? In the left hemisphere. Then what would have happened? Well, those fibers, if you were to follow them, what they are trying to see, the right visual field. So it was going to be the right homonymous hemianopsia due to this left hemispheric lesion happens to my optic tract, right? After that, one thing that you can see just from these drawings, whether the issue happened in the optic tract, right? Or if it bothers the entirety of the optic radiations later, is going to give you the similar problem. Why? Because these are the fibers I was following here. they took a relay from the thalamic nucleus, and then they were continuing back to the occipital cortex together. If I hit them together, it's no different than I hit them right here, right? But usually, what happens is that because these optic radiations take a separate pathway, we said the superior ones go through the parietal lobe, and then the inferior ones go through the Meyer's loops, will go through the temporal lobe. Usually their problems are going to be separate from one another, right? So what happens is, for me to get a quadrant anopia, what I need to do, I'm going to take 1/2 of the hemifield, then it gives me the quadrant anopia issue. If I hit the fibers right here, what's happening? Well, these were the ones trying to look into my left visual hemifield. But I'm only hitting, right, just these fibers. I'm not completing the bottom portions of these because those bottom portions are included in these fibers, right, in the Meyer's loop. So what I'm going to say is that if I hit these, go back to here, right, remember, the Meyer's loop, we're trying to see the superior visual field, like this here. So if I were to hit these fibers right here, the issue is going to be left superior quadrant anopia. It's not the whole health, but it's just 1/2 of the health in the field. Anyhow, once these Meyer's loop fibers, guys, really, it's very distracting, so sorry. Please do it later. I wasn't going to say anything, but it didn't stop, so okay. Anyhow, so the Meyer's loop will come back to the upper part of the calcarine sulcus. And what happens here, if they come all the way from the temporal lobes and hit the calcarine sulcus over here, again, you can just follow the fibers back, what they are trying to see, the opposite side, because these are just the temporal retinal fibers you were carrying backwards. Anyhow, once I'm here, right, what we said was, as we can see from the figures, they're all coming from both eyes. What I have to get this idea, the ocular dominance column, right? What this is trying to say, remember we said that there are these parvocellular, magnocellular layers that I collect the information and I map them as is into my thalamus and bring them over here. By the time it comes there, I still see them as contralateral, ipsilateral, all these 6 layers, right, neocortex. What has to happen, within each side, they sit right next to each other. I need to be able to make sure one column in my 6-layer area sees one eye, and after a very, very short period of time changes, I put the ipsilateral information. So contra ipsi, even though it changes still for these cells, because they see it in very close proximity to one another, it's going to be seen as one vision. Okay, that's, if you hear about the ocular dominance columns, that's what they are trying to talk about. So this is just, you don't have to learn all the details on it. What this is trying to show, right, magnocellular, parvocellular, retinal ganglion cell layers are going to be mapped as is into the thalamus as magnocellular layers just 1 and 2, and then parvocellular 3 to 6. If we just go back to this slide, this is the same thing that it was trying to show. Okay. And then basically this processing is going to start in area 17, and then I have area 18 and 19, trying to understand the form, shape, and also the color information on these things. Right? So there's a lot more information about this, but we'll just leave it like that. One last thing I want to mention is these vestibulo-ocular movements, right? What this is trying to accomplish, remember when we were looking into our inner ear, we said that this balance information has to be collected into cranial nerve number 8, and I need to somehow combine them with everything coming from my eyes, right? Well, what has to happen is that if I'm looking at an object and I still want to keep looking at it, but I turn my head and I still stay fixated, how can I do that? It needs to require this combination. What's going to happen, for example, let's say that there's an object maybe somewhere here, and I'm moving my head to the opposite side. What has to happen? My eyes need to converge themselves towards, stay focused on that object on this side. What needs to happen, if you remember your cranial nerves, for my left eye to move to this way, I need to abduct it, so lateral rectus muscle has to be active. So abducent nerve brings that information from that side. On my opposite side, right, the right eye needs to be pulled towards my nose, basically. So I need to activate the medial rectus muscle that is controlled by number 3 oculomotor nerve. So I bring it back a bit over here again. And then I need to combine the abducent nerve, oculomotor nerve, plus I need to know where my head is moving. Remember that acceleration, gravitational forces, etc. we talked about? That information needs to come from number 8. When I put all of these together, usually where I converge them could be vestibular nucleus or sometimes like superior colliculus, things like that. I'll combine the information together so that I can still maintain my eyes looking into the object, but I turn my head to the opposite side, right? This whole idea is called vestibulo-ocular reflex, basically. And it's going to be relevant when you have issues with like different bleeds, etc., especially bothering the oculomotor nerve. These people might have uncontrolled, like saccade-like movements, right? Sometimes if you go to neurology clinics, like when people are really under coma, you can sometimes open and you can see their eyes are constantly doing this movement, right? Because it's not really properly controlled. But anyhow, so if you hear about vestibulo-ocular reflexes, this is what they are trying to talk about. Yes? Well, it will come into the picture, right? But this is just showing that, in a sense, like sensory input, how it needs to be collected. Because all of these are not really doing anything. It's just like they are bringing that information that, yes, this one has to be activated, so I get that, but how I need to do, right? I collect basic sensory input, and then I decide, with the motor output to my extraocular eye muscle so that I can move it, right? So, yeah, there is also one small pathway, but it's not really the running the show, right? It's just one. Anyhow, so yes.
For this pathway, can you still be vestibular ocular movement if cranial nerve 3, 4, and 6 are, you can still do it, only 8 has to be impacted?
What can you say again?
So
No, okay, yeah, it cannot happen because what happens is, so you are thinking, right, I'm going to move my head this way, but I want to still maintain my eyes on that manner. So what I do, I put motor innervation to those through oculomotor, abducent, etc., right? And then I can abduct one side and then bring it towards my nose, right? So you basically do like this movement. So it still has to have the motor component again.
So if any of them are messed up, the entire thing is messed up?
Yeah, entire thing is messed up, yeah. Yes. So you need to understand what's happening with the sensory and then make a decision that I still want to look into this object and like move my head, right? And then the motor component needs to go to the extraocular eye muscles and control them, right? Okay, good deal. So I just want to show you like a couple common examples from this. The very commonly asked questions usually the optic chiasm, right? Because it goes back to the idea that whether you understand the neuroanatomy about this. So 62-year-old man comes to the physician because of persistent headaches and increased clumsiness. He cites repeated collision into door frames. Really, once you do your board questions like 2 years from now, you go through like UWorld 2,300 questions, USMLERX another 2,000 questions, ComBank another like 2,250 questions, right? I know, you will go through them. Once you go through them, what's going to happen? You will be, if you are good test taker, right? Even if you have no idea, when it says he cites repeated collisions into door frames, aha, it's the temporal vision is gone, right? Believe me, you will be like that. And blah, 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 his libido is over the same period of time. I mean, after evaluation, he's diagnosed with prolactinoma, right? I added this section, they don't tell you what it is, but they say like, oh, it's diagnosed with a tumor in the CNS. Okay, what is it? What they're trying to get is that, because remember the anatomy, pituitary gland sits right there with the optic chiasm. If anything, adenoma, something grows in it, it's going to push against the optic chiasm, and the fibers are trying to cross over are the ones going to be affected, right? That's why you are going to have this bitemporal hemianopsia. Both sides will be gone. But again, even though the boards are trying to get away from buzzwords that you don't recognize it, there are still so many of them left behind that you can really like get it. Collision into door frames, bitemporal hemianopsia. Okay, next one. So I don't know why would you get routine MRIs, but let's say routine MRI reveals the presence of a tumor situated in the left optic tract, proximal to the lateral geniculate nucleus. Okay. Again, good enough test taker, you just think, okay, left optic tract, proximal to the lateral geniculate nucleus. If it says left optic tract, it has to be right. And then what we said for this optic tract, it's going to give you homonymous hemianopsia. So then we keep reading, even though we know the answer now. The patient complained of having a reduction in his field of vision. The likely visual deficit can be characterized as what? It has to be right homonymous hemianopsia. Because those left optic tract is the ones trying to see the right side of the visual field on both eyes. One came ipsilaterally, the other one I crossed over, but they were still trying to see the right visual field. And because they're on the same side, I call it as homonymous hemianopsia. Little bit more tricky one, and I underlined this for you, left temporal lobe issue. This could be like after suffering an infarction of the left temporal lobe, or it could be that they will present you a seizure, epileptic case, right? Then you think that it's in the temporal lobe, then you think about Meyer's loop, etc., right? It could then take you to like second, third order questions that you need to know more things to figure this out. But anyhow, so after suffering an infarction of the left temporal lobe, this 52-year-old man develops olfactory auras that precede occasional complex partial seizure. Aura means that this sense of smell people will get right before their seizures. There are no significant post-ictal deficits. Post-ictal means after the seizure is over, except that the man suffers from anterograde amnesia. They are trying to tell you like this has to do with something in the temporal visual field or the temporal lobe. This stuff is passing through. Sometimes they will just tell you about a seizure and then they have an anterograde amnesia. This makes it a really good question because then you need to remember, okay, Meyer's loop, the ones passing through the temporal lobe, and then the Meyer's loop are the ones seeing my superior vision, and it also adds up to your knowledge that you remember those hippocampus type of like limbic cortex related memory areas are inside the temporal lobe, right? You know so many things already before you answer the question. Neurologic testing reveals that the man also has visual field deficit. What would be his, visual deficit? So even just by looking at it, right? So left temporal lobe, I'm thinking about quadrant anopia issues already, and this is in the left temporal lobe. It's going to see the right visual hemifield, because I'm only considering one part of these tracts, then it's going to be quadrant anopia. So right superior quadrant anopia is the answer for this question, right? So I know it's easier said than done, but if you just learn this figure, then you can answer any of the visual stuff, right? But of course, just to make it in a second, third order question, these are always going to be presented in like additional stuff that you need to recognize, all right? Perfect. So this was all I had today. Thank you for everything. And then I guess we can take a little break, a 10 minutes break, and then who wants to volunteer as the first group? She's up already. All right. Okay.
That's what you know what? You deserve it. You carrying that dinosaur around. Trying to like wait I think you already got it. Like you've already asked your parents. So we've been planning, and my cousin sent me the link for his like a shot. And I was like, we might as well.
We gonna meet with the group yet?
Yeah.
We gonna meet with our group yet?
Yeah. Exactly.
Did we? That's only me and you. We gonna have like gaps. It's gonna be a long time.
I'm gonna start. Kai's gonna take over after my point. So I'm done. I was gonna stand after that.
I think after Kaiser is me, right?
Yeah, go talk to him when he's gonna start.
I'm gonna go. Who's going after me? Who's even in the group? word.
Straight up.
Be like, all right, word. What are we doing for Thanksgiving?
She's like the youngest one out of our family. So most of our family is.
Should say something really awful.
No, it's a bench day.
Oh, well,
We grew up in California.
how's the family taking it?
It's just the first time I heard about it.
Yeah, well, the rest of them. That's how you follow that up. I want to read it.
She's smarter than I am.
I don't know what that means. Is it weird to Google that? Is that like the type of thing you don't Google?
I will.
Just at the end, be like, and of course. Oh my gosh.
Nancy. Sophia. I don't even know what I'm gonna say. I'm just gonna.
You got my name right. So there's a start.
I don't even know what I'm gonna say. I'm just probably gonna
You
go.
can be confused with this Nancy or other Nancy.
It's just because you guys are both Vietnamese, so I'm like.
Yeah, it's just thinking that.
Yeah. Are you gonna do
I'm
the?
doing alternative treatments.
Okay, I'm just gonna do the medication.
Regular treatments.
Just the medication treatments.
I'm not saying a word during my presentation.
It's nice.
I was like, is anybody good at presenting? Because your boys aren't.
You're not.
Not really. I mean, like, I can stand. I get nervous. Better in small groups. What?
Goodness, everyone has.
Exactly. I know my weaknesses. One of my weaknesses is being intelligent. Sex are not limited to sexual choice with regard to biological sex, gender, or gender identity. Sexual choice. Isn't that the same thing?
It's always what happens.
Isn't that shit just bisexual? Like it doesn't.
Yeah, it's all.
All the above.
Yeah, it's all.
Because I still get them. She's dead. What do I do? Tell Cedric to do I don't want to know what is happening.
This is why you don't get married in your 20s and have babies. This is what happens. Oh, that's it.
Julie, so Julie,
Julie.
so are we all like screwed?
I'll find out after Sterling talks to her.
Okay, Sterling, the one that's the representative to go.
Aren't you guys all gonna talk to her?
He offered, and I went to go.
It's really bad sign. Logan's gonna break up or something. Like a really, really.
To update us when he finds
Yeah, well, apparently Logan.
That's what I.
That biochem exam really ruined my day. Yeah, I think I did just below. Like
What,
one
you need
question
it?
below what I needed to get.
Dang. That's okay, man.
What's that? What does it
What are
sound
you gonna do? Ampera? 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 Ampera. Ampera.
Empanadas.
Fajitas. Julie, ask Sterling what Logan is up there so passionately arguing about.
How long is taking up the time? I need her to talk. I want to know what's up.
Apparently he was. Do you need a flash drive?
I got
You
it.
got it? Okay, cool.
I brought my flash drive.
Do you know why she got mad?
What do you mean?
Logan, why did she get mad?
So somebody was passing around the questions for the
They're like compiling all the questions.
In front of her face?
not in front of her face. Like somebody told her.
No, during the lecture, she was like, stop doing that. It's
Oh, I
distracting."
don't know what that was. That
Yeah,
was weird.
it caught me off guard. It's like, her voice was different.
Yeah, she's definitely angry with us right now. And has good reason to be.
So now they're arguing about the question compiling. It's over. They're going to her office. It's over already.
Is that what just happened?
It's over, man.
Right.
You don't gain anything. You don't stand to lose anything. You don't stand to gain anything.
This exam was going to be so easy. So easy.
You don't gain and you don't lose. And now,
Everyone loses.
yeah, now
Everyone loses. Because you can't listen for like an hour.
That's stupid.
It's so frustrating.
Like, if you don't want to be a part of it, you don't have to be a part of it. Yeah,
Like, shit, just tell us. Just address
Yeah.
the whole class.
What?
Y'all want to know.
So I'm
They're
gonna
all
fill that
tell
out at
us.
some point, probably tonight.
Maybe they don't.
She's not going to release the final grade.
Waiting for the T to spell it.
Unless a lot of people
She lost the T.
so that
She's just waiting to spell it.
Why is he not going to release the final grade? He has to.
he has to somewhere, but like not as soon as.
Huh?
That's why we need to delay it so we can challenge these questions.
Something's popping. And I'm going to sit back.
It's not mandatory, though, right?
Actually, it's water now.
No, it's optional.
Yeah.
And it's like unanimous. So.
I think it's an empty threat.
I still look forward.
What's empty there?
That is one of the classes I'm looking forward to.
Oh, I'm going to fill out all of it. And I'm going to give Nathaniel and Olga very good scores. And I'm going to hurt Palmeiras.
The problem
There were people, there's two people haven't volunteered in my group. I'm like, love it. Y'all go through
It's
it.
under endorsement.
that's why I wasn't going hard.
I will just type very clearly.
Yeah, time.
Yeah, just say it about him.
Yeah. Be like,
For the
hey,
section about the teacher.
Wilson did. Be like, hey, Wilson did great. She was very nice. He was like, Palmeiras.
There's a level of,
What now?
like,
I was like, this is less than under.
yeah, that's what I felt
Freshman
like.
year, maybe I would take this.
Like, I felt
I would not have
I
taken
felt
it.
like he treated us like high schoolers. With the way he was lecturing.
I want to hurt him.
Some people have bananas.
What is everybody printing out?
I kind of feel bad. 'Cause I think the way we kind of like growing up, she was like different from all of us.
You did brilliant.
Do you feel like excluded now?
Next, I wouldn't say. I think when she was younger, she didn't get along. Not
It really
get
just
along
turned
with us,
into a
but
big deal because they literally went to her office now to talk about it.
Yes. Her personality was just cool.
First of all,
What did she do?
I don't know the exact story.
She was doing a PhD in
a room.
in.
But yeah, apparently somebody
But
reported
now she's doing her
that
master's.
there was
Something happened. She
question
didn't go into
compilation
detail.
happening. So they wanted to say it's a violation of honor code. And then Dr. Oakham was like, she's gonna have to change the test, make it harder or whatever. And so I think the class officer wanted to talk to her about it, or she wanted to address and maybe find out the root who started it. There's a lot of cheating going on. Cheating with the whiteboards, you know, cheating with this thing. So it's like, it's just the fact that we're cheating a lot, so now they can't trust us.
Yeah, and people want to complain, like, oh, the administration's all over us. There's something. Yeah, so they're just right. Like, I don't get that complaint.
Like, oh my gosh.
When are you going?
Very passionate.
Do you know if that person got what expelled? 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 That person got expelled.